An international study has had promising early results showing that the venom of an Australian octopus could significantly slow skin cancer growth. One of the researchers behind the results, Professor Glen Boyle, joins us live now. He's the head of the Cancer Drug Mechanisms Group in the Cancer Program at QIMR Berghofer. Professor, really appreciate your time. Thank you so much. Before I get you to take us through the results, just start at the beginning here for us. What on earth made your team think that octopus venom could be helpful here? How did you even begin to hone in on that?
It's a very good question, Ash, and thanks for having me on. The research started at the University of Queensland quite a number of years ago, and we were fortunate enough to get a researcher over to QIMR Berghofer by the name of Maria Makapanomolu, and she brought a library of all sorts of different venoms with her. And of course, what do you do when you've got a whole library of different things and you arrive in a cancer research institute? You see what happens on cancer. So that's really how the research started.
Okay, so tell us about the results. What have you actually discovered about the impact of that venom on melanomas?
What we found is that the venom, which is a very small peptide, so it's just a little protein, if you like, was able to slow down the growth of a specific sort of melanoma cell. And these melanoma cells were the ones that have one of the most common mutations that cause melanoma by the name of BRAF. Interestingly, this peptide or the compound actually didn't do anything to normal cells, and that's why we really got quite excited about it.
And so, Glen, could it actually work on other types of cancers potentially?
Potentially it could, because BRAF is actually mutated in a number of different cancers, specifically colon cancer, thyroid cancer, small cell lung cancer, and as well as some prostates. So there remains a lot of work to be done. We can really open up now and see if we can find different cancers that this peptide will work against.
Yeah, a lot of work to be done for sure. What is the next step? What do you now do with this sort of discovery?
Well, the next step would be to try to make the peptide a little bit more active, because at the moment it could do with a little boost. And because we can actually synthesize it, we're able to do that in the lab, which is great. I think we should also test the compound against some more realistic models of cancers and different cancers that we have here. And so that would be the two major big steps.
Have you had interest from some of the big pharmaceutical companies? Do you start to get involvement from big corporates once you get to this sort of level in university research?
We haven't had any interest so far. It's very, very early stages, and I think big pharma usually get interested a little bit further down the track, and so we'll be working on that and try to advance the program a bit further.
Oh, well, best of luck with it all. Are there any other Australian marine animals you've got your eye on now? Do we have a lot to learn more broadly, do you think, about how venom from sea life could be medicinal?
Oh, absolutely. I think there's a great untapped resource in the natural environment. It's not just marine animals. It can be snakes, spiders, even plants. I think that we've got a lot to learn.
Professor Glen Boyle, it's fascinating stuff. We really appreciate you joining us and wish you all the best as you continue on those next steps now.
Thanks for your time, Ash.